now retired, as you can see. He retired, I don't know. So Chip spent 32 years working for the USGS downtown. Anybody who, in our class, has seen his papers. I gotta say, Chip he's not a groundwater modeler that sits behind a computer. I mean, he does that, but he also gets out in the field. It's one thing I've always very much appreciated about his work is he likes to get wet, get out in the field, and figure out good ways to find stuff out pragmatically. So I think he's going to touch on some of these things today. So there's the talk title.
All right, I want to thank Craig for inviting me and for Water Resources Research Center. I'm going to talk today about looking for wastewater effluent. Wastewater is kind of what goes down our toilets and out of our washing machines and so forth. People are interested in where it goes. Most of it will go to a water treatment plant and then somewhere injected into the ground through an outfall to the ocean. In the case of cesspools or septic tanks, it goes into the ground right where it is. There are processes that will attenuate the wastewater, bacteria. That's how septic systems and cesspools are designed. They bleed out their seepage, bacteria eat up a lot of the nutrients and organic matter, but some still persists. And the same thing with sewage treatment plants. They digest, eat up most of the stuff. There will tend to be residual constituents in the final treated effluent that end up being disposed. I'm going to concentrate on pharmaceuticals and wastewater indicators. These are things like over-the-counter drugs or prescription drugs that we take that go excrete from us and then out into the waste stream. And the wastewater indicators is a large class of about 45 or so compounds on our analytical schedule that include everything from fragrances in your perfumes and colognes, soap, constituents, just a whole bunch of stuff. Mosquito repellent, a lot of times is found. Caffeine from coffee is sometimes found. All right, so let's move on. So as I say, these projects are driven by interest in wastewater fate by several government agencies here in Hawaii, NOAA, US EPA, Hawaii Department of Health, National Park Service, and USGS. I'm only going to talk about the projects in yellow here. There are several others that I'll bypass, but there are either reports for them or we've got a couple of the surveys posted on our webpage at our USGS website. And I'm going to jump around too. I'm going to get a little out of sequence on time. So Craig invited me to speak to his submarine groundwater discharge class, and so for people not real familiar with the topic, here's a generalized diagram. Up here it rains, you get surface runoff from streams, water infiltrates into aquifers. It'll flow through the aquifer and discharge at the shoreline. This also shows a confining unit for a deeper aquifer that'll tend to shunt most of the discharge out here some distance offshore. And you can have various combinations of that depending on where you are. Here's a pretty simple-minded survey, a weighting survey to look, try and detect groundwater discharge and wastewater. In the inner tube here, we've got a multi-parameter water quality instrument. We've got a GPS unit. We've got sample bottles. Every certain distance, we grab a bottled sample for laboratory analyses. This is at Kualoa Beach Park up here on the windward side. It was closed at this time because of contaminated water. High fecal indicator bacteria counts cause them to close the beach periodically. And yet people don't pay it too much mind if it's a sunny day. The problem, again, high fecal indicator bacteria counts were causing beach closures, and there are beach park restrooms every certain distance along in the park, and each of them has a septic system. And these were thought to be the main contributor to the problem, and Department of Health had been monitoring marine water, shallow recreational marine waters for fecal indicator bacteria for quite some time. The bacteria sampling was their main tool at the time, probably still is. And they had resorted to frequent pumping out of these systems. Here's one of them. Here's the truck coming to pump them out. And so how might any of the effluent from here get into the ocean? Well, if it rains hard enough to pond, maybe there's some overflow in the tank and direct runoff, or maybe some of the effluent seeps through the sand aquifer and out to the shore there. So these were some of the starting working hypotheses. But there are non-human sources of these fecal bacteria too, and I think the general test for it doesn't necessarily discriminate. There's a bunch of seabirds in the area, there's a large feral cat population in the park, and what do we got over here? This guy, dog on the beach probably. For a while there was a flurry of interest about turtles because there were clods of something washing up on the beach on the windward side and thought to be maybe human fecal matter. I think it turned out to be turtle fecal matter for the most part. So people were kind of running around wondering how to discriminate some of this stuff. We call what we were doing a multi-tracer approach. I list a bunch of them here. They're what I call ambient or inherent tracers. The other type of tracer, like Craig's group did, is what's called an introduced tracer, where you might take rhodamine dye and put it down a well and look for it off the shore. But the philosophy here is to test for things that are already in the water because they might indicate something. Salinity is a fundamental measure, and that'll give you a measure of fresh water versus seawater. Several simple nutrients, nitrate, ammonium, orthophosphate. These can indicate human animal waste, but also fertilizers. Nitrogen isotopes, stable isotopes of nitrogen, can start to discriminate between waste sources and soil nitrogen or fertilizer nitrogen. Stable isotopes of boron can tell you if you've got a large amount of home laundry detergents, because a lot of them tend to contain boron and have a very light delta 11B signature. UV fluorescence. Many of these laundry detergents have optical brighteners to make your shirts whiter and brighter, and you can look for those with the fluorometer and or black light. A lot of these can be somewhat ambiguous and not real specific. A few that are more diagnostic are pharmaceuticals. They'll be a pretty direct indicator of household wastewater. This wastewater indicators, analytical schedule I was telling you about, same thing. And then there are what they call bacterial source tracking methods. These are looking at the actual DNA in the bacteria that they collect and discriminating what the source is. This is a quick slide about the nitrogen isotopes. Under the right conditions, they can serve as a good tracer of nitrogen source, but there can be ambiguity. Here's the delta 18O in nitrate. Here's the delta 15N in nitrate. And through various studies, they've found that results tend to fall out in a few different fields. Natural soil nitrogen, like from a forest, falls about here, kind of 3 to 9 or so on the nitrate scale. Nitrate fertilizer tends to be lower on the nitrate scale, higher on the oxygen scale. Manure and septic waste, you can see, occupy a large field. It's coincident with the soil nitrogen, but tends to be a bit heavier, like on the order of 10 or so. And then denitrification processes can take nitrogen anywhere on here and drive it up in this direction at a slope of about, I think it's 2 nitrogen for every 1 oxygen. So that's kind of the framework of how the N15 is used. This is our first platform that the Hawaii Department of Health guys rigged up, an inner tube, a bucket, an adjustable depth thing here for our instrument. Here's our GPS, and we would toss sample bottles in there. Later on, we went to Maui. We didn't ship that thing over because it was heavy, so here I just strapped the instrument between 2 coolers and off down the beach waiting like that. Also in Maui down at Kihei, eventually we put it at the back of a kayak and had one of our investigators kayak down. That was a bit faster. And these are the parameters that are on the sign: salinity, specific conductance, temperature, pH, dissolved oxygen, turbidity, chlorophyll A. So here we are back at Kualoa, and you'll notice that we're quite close to shore. There's several reasons for this. One is that in the literature there's a model that people follow that recognizes that there will be wave slosh up the beach face. And that'll tend to be salty water that'll tend to re-infiltrate and have a return flow here. And that if you've got inland groundwater coming along, that'll tend to go a little seaward of that. And then out here you get ocean water that's circulating into the aquifer and coming back up with some of that fresh and brackish water. Here's a nice model of it showing the same principles. But the main reason I was here is that a couple of years ago I was up at Sunset Beach on a calm day, sticking my toes in the sand, and it was nice and cold right on the toe of the beach like this. So we did one pass down here and then the second pass either just inside these blocks or maybe even outside, I forget. Here's a result in terms of salinity. It's a funny scale here. I'm expressing it in percent seawater, going all the way from near 100% as the hot color down to as little as 92.5% in blue. And it turns out that we got our freshest water up here in this little sheltered cove where the shoreline jogs out like that. As much as 7%, 7.5% fresh water here, just by wading along. You can see that most of the extent of the park here is pretty close to seawater. We're not really detecting much freshwater discharge there. Intermediate results elsewhere. So this was walking before you run. This was baby steps to make sure that we could track along with the GPS and get the results into the computer, display them up. And we could display all the other parameters on the instrument as well, just showing you salinity. The other thing we did was beach pore water sampling. Here we've driven a steel well point into the beach and pumping it out and then into sample bottles for more sophisticated chemical analyses. This is the hard way to do it. We've got a hand drive hammer drill. We're putting them in. This is a bunch of the Department of Health guys that were participating. And this is really hard. Here we're trying to get up the beach face far enough to be out of that swash zone and to hopefully collect some groundwater that's coming out in the shallow superficial sand aquifer. If I were to do it again, I'd be trying to hook up with NOAA to pull this puppy out. It would be much easier. Here are the results for N15. Well, it turns out we got our heaviest, our nitrogen that's most likely to be indicative of wastewater up here at that cove where we have the good freshwater signature. And pretty light, on the order of 5 through most of the park. Intermediate results here, 7.5, 8.7, 9. So we kind of revised the thinking to not only be concerned with these septic systems, but also perhaps non-point pollution from across the highway. It's Kualoa Ranch. They've got horseback riding. They've got cows in the pastures. And these houses up here that are on individual cesspools. This is a look from the park back up to the mountains. If you get a lot of rainfall, it's going to come down these chutes, infiltrate into alluvial fans, colluvial fans at the base of the mountains, down into the ground, out through the sand aquifer and onto the ocean. And this is a stormwater swale during fair weather here. When it rains hard, those will fill up and be a source of infiltration and flow through that shallow aquifer as well. What I'm driving at here is if you put a groundwater hydrologist out on the landscape, they'll observe some of this stuff and go through a certain type of thinking and so forth. So for an SGD group, you might like to have a groundwater hydrologist collaborating, just to get some of this kind of thinking introduced. You can see this water looks kind of mucky. Now whatever's in that is going to be going through the aquifer, minus whatever attenuation might be happening in the aquifer. Here's looking back the other way. This is after a big rain event. The swale's nice and filled. And I did some retrospective hydrology. I went back and looked at stream gauge records and rain records and found out, well, gee, around the time of these persistent beach closures, we had a couple of big rainfall events. And I was able to look in the newspaper on the website and came up with this picture. This is one of the bridges out there nearby. I don't know if it's Hauula or where this is, but this is a pretty big rainfall event. And this, turns out this happened 4 days before beach closure that had one of the highest counts that was in the record out there. And so I'm pretty convinced that this particular beach closure, and a few of them after that, resulted from what's now called a groundwater event, which is a lot of runoff, muddy water, all the dog poo and stuff from everyone's backyards goes out. And these days, your Department of Health is telling you on television, don't go in the water. Back at this time, they were just starting to give that kind of advisory. So there can be a number of other events out there on the landscape besides a wastewater source that you might fixate on, like septic tanks, like injection wells. This was the year prior, in 2006. I think this was one of those years that we were talking about 40 days and 40 nights of rain. For those of you out here, you might remember. This is just north of Kualoa Park on Cam Highway, entirely flooded. Here's Kualoa Ranch over here. I don't know if water's going off the road into the pastures or vice versa, but there's direct runoff here. There's infiltration into the superficial aquifer. So that's an example of non-point runoff and some of the goodies it might carry. This is a project over in Kihei, Maui, and this did have to do with injection wells. I came up with a three-dimensional model of the injection process, what the injection plume looks like, and this was with NOAA and collaborators here at University of Hawaii, Celia Smith's group, the experts in algae. This was driven by concern with algae blooms. There had been concern all the way back into the 90s when there were large algae blooms over on Maui. And there was a series of studies back in the 90s by NOAA, EPA, Department of Health, and a bunch of consultants and other researchers to look at the problem. They came up with some answers back then, but they were dissatisfied with how specific they could get on the wastewater sources. So come the 2000s, we're still looking at it. This is from Celia Smith and Jennifer Smith's report in 2000, where they were looking at algae over there. And this is a photo of Hypnea, red algae. It's an invasive algae over there in the Kihei area. It washes up on the beach, it rots, the tourists don't like the smell. They have to come out with a tractor and drag a bunch of it off the beach. It's kind of a mess. This is Ova fasciata. This is an indigenous algae, but it's still no less onerous if it starts to rot. And this is the two types are kind of all mixed together and really a thick accumulation of it. And this is at Honokawai, north of Lahaina. And interest centered on these areas, Kihei and Lahaina, because it's known that there are wastewater treatment plants there. They treat the water, polish it as good as they can, but then it goes down injection wells. It is higher in nutrients than the ambient groundwater around there. And so people have long suspected that these are a main cause, or at least a contributor to some of these algae blooms. So this is where I worked with my model in 2006. This is actually the outline of the model grid. There was an earlier model up at the Kahului plant in 1977. That's, 30 years before computers were really in their infancy back then. And then more recently, there's been a USGS model for the entire Lahaina regional area by Steve Gigrick and John Engott. And they didn't really look at the injection process in a lot of detail because they were studying a larger area. But they did do a couple simulations, what-if simulations if the injection were ceased and so forth, mainly showing what the result was going to be in terms of water levels. But I took a closer look at the injection plume itself, and this is the starting point. You envision a coastal aquifer. This is kind of a block diagram, but it's also a three-dimensional block output from the actual model. But stretching back into the uplands there, you've got what we call a freshwater lens. This is the water table. Land surface is a couple of 100 feet above you. It's been stripped off. There's salt water down deep, and then there'll be a mixing zone between the two waters. And it turns out that the fresh water flows out based on its, basically the slope of the water table. Gravitational gradient is driving that. And the mixing happens, and it actually entrains salt water and discharges it down here at the shore. Here's where all your discharge is, at the shore, close to it. It actually sucks salt water into the aquifer to make up that volumetric depletion of the salt water there, so you get this recirculating salt water. The yellow dots are the grid points in the model. You can see there's a whole bunch of them in this area. This is where we were going to inject, so we needed detail in the mesh here. And out here, there's just a vertical wall. That's not realistic, but because of the type of simulation I was doing, steady state simulations, it ends up not being a factor. Here on southern Oahu, this more gently sloping surface goes out for miles and miles and miles. Let's see. So here's injection. So folks that want to engineer these injection plants aren't dumb, so they want to inject it kind of deep. In this case, the top of the injection well is not open to the aquifer until 50 feet below the water table, and then it goes down to close to 200 feet. So that's an attempt to push the stuff deep and let it go somewhere. The colors here, 3 to 20% salinity in blue, and then 40, 60, 80, 100% seawater down here. I've stripped off everything 3% or less salinity. And one nice trick that makes is it leaves you with a hollow core to the plume here. It helps to visualize it. So at any rate, we try and inject it deep, but it's buoyant because the effluent is only 1% salinity. You get in your shower, that's fresh water. You flush your toilet, that's fresh water. You do dishes, that's fresh water. So it's much less dense than seawater, and if you try and inject it into seawater or denser brackish waters, it bobs right back up to the top and flows out. So now you're going to have it shallow right along the shore, right where you don't want it. But that's physics. And we've known this since the 1970s. Frank Peterson and his group of researchers at Water Resources Research Center started out doing various sandbox models and thin plate models where they would have fluids between two thin glass plates. And they came up with results like this. They documented the buoyant rise of the effluent and that it flows on down to a conceptual shoreline down here. It doesn't come all the way to the top in these cases because that'll depend on the volumetric rate of the injection compared to the ambient flow that's going there. And I can only presume that they had a pretty strong ambient flow compared to the injected flow, so that it stays down here at the bottom. In plan view, there were things like this in the textbooks back in the 60s and 70s. This is an example from John Mink, renowned hydrologist here in Hawaii. This was from the Guam Water Resources Research Center that had just started. It was the first report in their technical report series. And the idea here is that if you have a natural flow field of groundwater coming down, you inject, that this injected water will displace the ambient water out to the side, and the yellow here actually outlines the dividing line between the ambient regional water injected effluent inside of the plume. Now the width of the plume here at the coast will largely depend on the volumetric rate of the injection versus the volumetric rate of the ambient regional flow. If your injected volume is small, you'll get a narrow plume. If it's broad, you'll get a broad, if it's a large volume, you'll get a broad plume. So there was a lot of these basic concepts known as early as the 60s and 70s. Well, this is a result I got at Kihei. So back here with recharge up in the forested area where it's rainier, upslope, we do a water budget for the area. We end up with a volumetric ambient flow of about 3 million gallons per day per mile of shoreline. And you'll notice it's not expressed in meters per second because in our work we like to express in units that people can visualize and are familiar with. And all of the water resources guides in Hawaii talk about million gallons per day. They estimate aquifer yields in million gallons per day. On an atoll that I worked out, I got to see what a million gallons per day, or a million gallons looks like. It's a 10-foot tall tank, concrete tank, 130 feet in diameter. So early in my career, I got to know what a million gallons looked like. And people in the English-speaking system know what a mile is, so these are the units we used. Now, we inject 3 million gallons per day of the effluent at Kihei. Well, it turns out the injected effluent kind of sticks its elbows out enough that it ends up commanding just about a mile of shoreline in terms of its aerial extent. So that's an outcome that squares with common sense if you think the process is through in that way. What else was happening here is there are a few houses on the upcountry road here near Kula, and so forth. There's some small farms, some pretty minor ranching. Between there and down here is all scrubland, dry forest, Kiave, Halekoa, that kind of thing. And then down here in the coastal strip, we've got resorts. Right on the shore, we've got condominiums, we've got individual households, things like that. Following that thinking, if you're interested in nutrients, you might express nutrient loads. Well, within a certain volumetric water flux, you can measure the nutrient concentration and divide one into the other and end up with kilograms of nitrogen, if you like, or phosphorus. And because you're concerned about algae blooms from maybe too much nutrients in the shore zone, you pursue that kind of thinking ways and some computations. And this is just to point out that there are different types of nutrient loads. Someone might be inclined to focus entirely on this wastewater aspect, but you've got to recognize that there are other sources of nutrients out on the landscape, and this simplistically illustrates some of them. From the forest area, you'll have a certain amount contributed by vegetation decaying. If you have some ranching with cows, they'll be making their contribution. Here's the point source wastewater source, and I call it attenuated wastewater because we found that when you inject these wastewaters into the ground, bacteria will get busy and chew up a lot of the nitrogen in these plumes in particular. And what you end up discharging at the shoreline is much attenuated from the concentration that went in. What else? Then we get down into the urban strip here, and you've got urban fertilizers. You know, people like to keep their lawns and their landscaping healthy, so they'll apply some fertilizers. We've got cats and dogs in the suburban landscape. They'll make their contribution. There's one particular housing development down in this area called Maui Meadows. It's a bit upland, and they're all on cesspool. So that's a different potential contribution than most of the rest of the strip, which got on centralized sewer lines that go to the one centralized treatment plant. All right, so just some very simplistic nutrient load thinking to arm you to carry on further. Well, then we got an opportunity to take these wading surveys that you saw earlier back over there to Maui, revisit the Kihei area where I had done the modeling, and then also to get up to the Lahaina area where there was a bunch of concern about the injection, the wastewater treatment plant and injection wells up here north of Lahaina at Honokawai and what they refer to as Kahekili Beach along here, or what's the resort? Ka'anapali, North Ka'anapali Beach. It's also called an airport beach because there used to be an airstrip here. Small planes would fly in. And this was in conjunction with Hawaii Department of Health and US EPA. And this was funded through their, the Beach Act under EPA that they get funding for to ensure that recreational waters are fishable and swimmable and that sort of thing. And the Hawaii Department of Health administrates that program in Hawaii on behalf of EPA and can get funding from them to do this kind of stuff. So here we are back at Kihei, and here's my plume. So this is actually a three-dimensional solid. The pink line is the extent of it at the water table, and outside of that, we're actually seeing the outside of this bulbous shell in the subsurface. And so I stripped that out and stuck it on here in my GIS system. And this is what we get. There's a park down here, Kalama Park. That's kind of where it's centered on. And these are, again, the 20% bands. This time not in salinity, but the actual effluent. So at the center here, we've got like 100% effluent down to 80, 60, 40, 20, like that. And it's all discharging down here at the shoreline. The data points are N15 in algae that are growing on the reach, along the beach, and on the bottom in the shallow offshore. These samples were collected by Megan Daler and Darla White from the University of Hawaii. This actual set might have been just Megan, I forget. Megan's done a lot of work with Craig's group over at Kahekili, and I think Darla works for DLNR as part of their aquatic resources group. So at any rate, one thing that happens is as you inject effluent with a N15 value of 15, it goes in the ground, and some of it emerges at the shore, very little changed, but we had one well on land where we got a value of 23, and that's indicative of that denitrification process on that graph before that will drive the N15 heavier to larger positive values. So this was a first documentation that we do get considerable denitrification of nitrogen in the aquifer. Although again, at this site, it looks like a lot of it comes down and gets out without changing much. We have to keep in mind that this is a one-time sample. We don't know the variability of that from day to day or seasonally at the plant. These are one-time samples, but I'm pretty confident, at least on the algae, you get similar results. The idea there is that the algae sit on the bottom and they like nutrients to grow, and they'll happily munch nutrients and incorporate in their cell structure, and let you collect the algae, dry it in an oven, powder it, and run it through the instrument to get your result. Notably, there was a nice submarine spring that we detected over here. There's a pool in the break wall between some of the boulders that would nicely accumulate the water before the waves would mix it up too much. And down here at the south end, there was a nice beach spring that gave us, tapering off to a low value, similar to what we were seeing as background on land. And so we were pretty happy that these results kind of confirmed the results of the modeling. They're pretty consistent. Think about this spring. You saw later that we were using a kayak, and so we'd move a little faster, and eventually the UH guys, I think, or USGS guys were using a jet ski to go even faster with instruments. it's fine to go fast and do some mapping, but you also want to take at least one wading transect slowly, because we came upon this spring only by wading, and it was a nice roiling spring that would roil the surface of the water in kind of a bubbling rate at waist level. So that was really nice, and we certainly stuck our sample bottles right in that spring. So go slow for at least one survey to find what's on the landscape.
Chip, could I ask about that?
Yeah.
The orientation of the plume.
Yeah.
It matches up extremely well with the N15. So is it actually perpendicular to slope, or is the plume been adjusted at all for your geochemical?
No, all the modeling was complete and published long before we did the survey. And yeah, everyone's been struck by the southwest orientation. Why not more straight out? Some of the possible answers to that are, well, you have a re-entrant in the coast here, so that might tend to orient it that way. The downslope direction is pretty much straight offshore, as you would expect. The other thing is that as you're coming down up on the mountain axis, you're getting drier and drier as you're coming down to where it plunges off into the sea. And so maybe the recharge is greatest more up in your northern corner of the study area than it is farther down. And that may also provide some bit of reorientation there. But no, there were no aquifer properties. My mesh was oriented straight offshore, my modeling mesh. Yet this was the result that we came up with, so it did what it wanted to do. In technical terms. Okay, this is a weighting salinity result. This is with the instrument. And here we've got a different scale now. I flipped it and I'm expressing it as percentage freshwater fraction. So blue is going to be close to seawater. That's 2 to 5% freshwater. And we got up to as much as 25 to close to 50% freshwater measured by the instrument in these hotspot areas. Again, that pool that helps sequester the water in this submarine spring. A little hot spot up there. This is Maui sunset. That was where we saw the pictures of the heavy algae accumulation before. And notably, that's a good mile and a half north of the injection. So from those photos, you can deduce that you don't necessarily need a wastewater injection source to generate algal blooms. Okay, over to the Lahaina side. This is the Kihei beach area. Here's the beach park. Up here is the waste treatment plant, and that's where the effluent is injected. Our sampling was not blind. It was guided by advice we got from Megan and Darla, who had told us that, well, for a long time, scuba divers have been interested in these prominent seeps here. You can see water bubbling up, and it was warmer than the ambient water. So people had been interested in these, and Megan and Darla had actually done prior algae surveys that showed a nice gradient with heavy N15 here. So we were basically following up, duplicating their work and confirming it. These are water samples. We collected 12 dip samples at various places, wading along. And Megan and Darla put on their scuba gear and dove under and did the collection at the seeps for us. And this is targeted sampling. We're not going out blind and covering the entire area in a mapping sense or detection. I'm sorry, we do want to detect, but we're not. We're targeting to areas that we already know. We're not going blind. This is how they were doing it. An inverted funnel would concentrate the flow up into a bottle. In this case, it's a plastic bottle, but I was sending them down with these nice, clean and baked glass bottles that we need for the organic constituents analyses. And they would take them down and uncap them and then fill them for us, cap them up, and bring them back to the beach where I was sticking them in a cooler to isolate that groundwater discharge, because we're interested in what's in that, not what's in the seawater. And here are the results. Now, these analyses are very expensive. They're like $1,000, $1,500 a pop. There's 2 analyses, so it's going to be $3,000 a site. So we didn't even analyze all the samples for these constituents. We did nutrients and cheaper stuff at the other sites. But we only analyzed the 2 seeps. Looks like one site up here that we might have expected might be getting out of this concentrated area of interest. One sample up by where the stream comes out, although it was not flowing, it was fair weather. And this is the effluent that we sampled, again a one-time sample on one day. But we ran it through this pharmaceuticals analysis, and there's about 20 or 20 constituents on the schedule. The only ones we detected were at the treatment plant, carbamazepine sulfamethoxazole. This is an anticonvulsant for epilepsy patients or sufferers, and this is an antibiotic. Well, it turns out at both of the seeps we got both of those same constituents, carbamazepine sulfamethoxazole. Additionally, here at the North Seep we got diphenhydramine, that's Benadryl. And then up here, we didn't get these, but we got caffeine at both sites, and 1,7-dimethylxanthine turns out to be a caffeine metabolite in the body. You metabolize caffeine to some extent. Some of it will break down, and that goes off in your waste stream. Again, not identical in time. Anything that we detected being injected that day differs from results here, where the source waters would have been injected months ago, probably, according to your tracer survey. Turns out that the carbamazepine and sulfamethoxazole are found all over the U.S. most frequently because they're very persistent to break down in the treatment steps and so forth. Caffeine tends not to be found in treated effluents like this because there's what they call an activated sludge stage where stuff sorbs to solids and bacteria break things down in a particular way that caffeine tends not to get through that process and out into the final effluent. So why are we getting it here? Well, I can only speculate 2 possible causes. One is directly from swimmers, because there are big hotels and resorts along here and a lot of people swimming. The other possibility is seepage from leaking sewer lines. And this is an important topic Craig and I talked about today that he's pursuing in more detail lately. For all the resorts, they'll have sewer lines that will collect their waste and send it off to the treatment plant. Well, sewer lines in the ground are notorious for leaking. When it rains real hard, the water table rises and they get a lot greater flow to the treatment plant than the normal waste stream. The wastewater specialists, the piping guys, have found that, well, you raise the water table up, the pipes aren't a perfect seal. Water leaks in, and you get a lot of stuff that goes, you create a lot more volume being sent to the plant during really heavy rainstorms. Now, what happens in dry weather? Well, the water table drops, stuff goes out of the pipelines into the ground. So wherever you have sewer lines, you can be leaking waste constituents directly out to a nearby shoreline. That's the other explanation for the caffeine. The other schedule we looked at is waste indicator compounds, and these are a bunch of the soaps and so forth, perfumes and things like that. At the injected site in the effluent there, we detected bromoform, which is a disinfection byproduct. If you take a wastewater that's rich in organic matter and you chlorinate it, and if it's also brackish with a certain amount of seawater salinity in it, you'll have enough bromine that hooks up with some of the carbon and oxygen and makes bromoform. The other one that's commonly found is chloroform. So we detected bromoform, 2 particular fragrances. They've got long, detailed names. 2 particular fire retardants, and then a bunch of others, and maybe 12 or 16 things we detected. Well, out here, the one seep sample that we analyzed, we got the bromoform, the same 2 fragrances, and the same 2 fire retardants. A lot of clothing these days is treated with fire retardants, particularly for kids and toddlers, because you don't want them getting next to a candle and going up in flames. So when you launder those clothes, the fire retardants wash out, and they're another type of constituent that you can pick up with this type of sampling. Let me check the time. Okay, I better wrap up quickly. Here was plume mapping with the N15 and algae, again collected by Megan and Darla. Well, we got light values about 5 to 6 up here, coming up to heavy values up to 36, 38, 39 over here, tapering off down here, and an upland well, a value of 1. I take that to be indicative of former agricultural fertilizers. This area used to be in sugarcane 20, 30 years ago. A lot of that's probably still in the ground. 23 at the effluent, and so denitrification driving that heavier by the time it gets out here. Craig's group has much more interesting results after this. Well, one question arises, well, why hasn't this stuff gone straight out to the shore here? And again, here's where a little geologic thinking comes in. Here's a stream valley. What happens during Pleistocene low stands of sea level, like as recent as 17,000 years ago, sea level is 300 feet lower, and these valleys are marching out across the coastal plain to far offshore. As you raise sea level back 7,000, 6,000 years ago in the Holocene back to near current levels, those valleys will drown, and streams coming out will drop their cobble load, their boulder load, right in the water, and all that will come out is kind of muds offshore. Well, pretty soon you build up a delta of what's mapped as alluvium on the geologic map, in pink. This is basalt in green. And it'll be boulders, sand, mud, muck. Not very permeable compared to the lavas. Well, the other thing is you've cut a valley into the lava aquifer, and now you filled it with this mucky stuff. That's going to be a barrier to groundwater flow. And, one of our colleagues at USGS has written an entire report about this. The early guys like Stearns and Wentworth focused quite a lot on these valleys and what their effect was. So I hypothesized that, well, there must be a paleo valley fill in here that's preventing this effluent from going up there. This well just happens to be on the south side of that valley fill, that valley barrier, and that's got to be forcing everything down in this direction. Well, offshore in the bathymetry, there's a re-entrant here, and guys that are surfers know that that's going to be your surfing channel. When the waves are big, you'll paddle out here, and the waves will be breaking here, not so much here. There's a good chance that this is the probable course of this paleo valley. So at any rate, this is a hypothesis that the valley fill diverts the effluent, and later modeling by the Water Resources Research Group with Ollie and his group verified that. I don't think they could get their model to work correctly without a barrier there. Let's see, moving on real quickly. Oh, this is in the same area. In the 90s, they tried a dye tracer test. I'm pretty sure it was rhodamine. This is Tetra Tech out of California, funded by EPA. They were thinking, okay, inject here, it goes out here, so they established a grid offshore, and they injected it, and then a month or two later, they started going back and forth with a boat, with a tube all the way to the ocean bottom, sucking water up through a fluorometer. And they were also thinking, their other hypothesis was that maybe there was also a confining unit that might make most of the discharge happen some distance offshore. At any rate, here's one of their results. Now, down here is where those seeps are, in this lower left corner, and here's their results, contoured, parts per billion tracer, I guess. And there's a lot of tight contours down here in that lower corner. If you tilt it that way, it would be oriented. They didn't pay it a lot of mind. They remarked on it. They said, yeah, divers have told us about seeps, and there's an area here that we're a little puzzled by and should get more attention next time. But I think they had it. They just didn't know it. They didn't give it enough credence because they were expecting flow up here and maybe offshore. I think they tentatively were afraid that they missed it because it was farther offshore. This is wading salinity at Lahaina. Blue is salt water, not much fresh water there. Intermediate fresh water here around the seeps, and then a real hot spot down here. Well, what's this about? Well, there's a little laguna mouth back here. I came to call it Laguna Verde because it's a little channel of water here, and it's pea soup green with a little phytoplankton bloom that's in it all the time. Up here at this end, this looks like a silt delta to me. There's runoff swale. So what happened here is you want to get on Chip Fletcher's coastal geology page. They've got historical air photos. Back in the 50s, this was all sugarcane, and up here on the hill at Black Rock, there's a big fuel tank and other stuff to support the sugar industry. And this was built, it's a revetment to pull barges into, and I think they would load the fuel and stuff off and then maybe load up some of the sugarcane product out elsewhere. Well, in the 60s, they made a mega resort through the whole area. And this is how they landscaped it here. In one of the photos in 1960 or something, you can see big earth movers moving around, grading that area. So that's a result of stormwater engineering. For development, they'll put their engineering smarts together to get water off the landscape so it's not flooding your homes. So here's Boca, Laguna Verde, the mouth of Laguna Verde. Here's the swale in that area. And up here, I'm pretty sure this is a storm culvert because you've got all the terrain malca of there. It's got to get rid of its water. So it probably accumulates in ditches parallel to the highway, just uphill. And they route it under the highway, and the storm culvert comes down through this swale and then out. So if anyone wants to look at surface runoff processes and nutrients delivered by those processes, this could be a cute case study. This is one of the later things. This is over in Kona. Department of Health said, hey, people are squawking about another treatment plant and injection over in Kona. What can you do real quick to tell us about it? Turns out they don't even have an injection well there. They treat it. It goes in a pipe over here and goes in a collapsed lava tube open pit in the ground, and out of sight, out of mind, off it goes. So people have long wondered, well, what's happening with that? Is that a problem? It turns out there were a couple of monitor wells put in for a proposed development here that didn't really come to fruition. So we were able to get on the landscape there and take a sample at the plant itself, and then a sample at a couple of down gradient monitor wells and 2 springs in the back wall of the harbor. And we analyzed those, and here's our friend carbamazepine, detectable at the plant effluent, detectable over here. Higher, which might be puzzling, but again, these are not coeval in time. This could result from something injected with a higher concentration sometime in the past. And down here, we were still detecting it. The E-value means estimated, and this is a peculiarity of the way our lab reports. It's actually below what they call the typical reporting level, but they're confident it's there because they get a blip on the gas chromatograph in the right place, and they get the right breakup fragments on the mass spectrometer for that compound. So the other way that they indicate it sometimes is present but not quantifiable, and other commercial labs might indicate it as trace. But we still consider these detections, even though very low concentration. These are in micrograms per liter, which is parts per billion. These are less than a part per billion, so you're talking parts per trillion now. So this would be 86 parts per trillion, 130 parts per trillion, 28 in 1 part per trillion. Very, very, very vanishingly small trace levels. We can detect it. There's not many things that are going to have much of a ecologic effect, those parts per trillion concentrations. Here's sulfamethoxazole, same thing. The only place where we got an absolute non-detect was at the second spring. And here's optical brighteners. We had a little handheld fluorometer that, again, make your shirts whiter or your clothes brighter. We would put samples in, and if there's fabric brighteners from the laundering gray water into the wastewater stream, you get a detection. Our tap water control was a value of 1. These are just meter readings, declining towards shore. You can get interference from dissolved organic matter. There's plenty of organic matter in the waste stream. So you're not necessarily certain that you're getting the actual fabric brightener signature itself. There's a couple tricks you can play. But it's a reconnaissance tool that can be useful. Craig and Adam Johnson worked in the same area, sampling wells all over the uphill landscape and waters from the harbor, and they mapped out an entire field of phosphorus enrichment off of a seawater to upland wells mixing line. So this indicates a lot of excess phosphorus in the harbor. It turns out that that treatment plant was higher in phosphorus than most other effluents I've sampled. And the temperatures of the wells that we sampled, interestingly, fall along a mixing line from, well, the effluent at 24 Celsius. I just connected that with subthermocline seawater that OTEC pulls up in its big pipeline a few miles north at basically seawater and about 6 degrees Celsius. And lo and behold, all of our samples fall pretty close along that line. Steve Balls, a consultant on the Big Island, hydrologic consultant, had sampled, taken a sample from deep in a well and came up with this value. And this is a salinity profile, no, temperature profile. Water temperature down here, 16 to 22 Celsius, and depth below water table. This is a well that goes about 120 feet deep. Well, it starts out at about 21 degrees up at the water table and kind of plummets until it flatlines at about 7 degrees Celsius. That's pretty damn chilly for Hawaii. I recall that 20 Celsius is about 68 Fahrenheit, and that's chilly swimming on the east coast of the US. So there's 20, so that's going to be 62 Fahrenheit, maybe 60. Chilly. So what's happening? Well, over there in this area, our freshwater or brackish lens is underlain by cold seawater, which is unusual. Most other places in Hawaii, you get down into the seawater, it's warmer than the freshwater because the freshwater recharges up in the mountains where it's cold. So typically your freshwaters in Hawaii will tend to be about 19 or 20 C, and the salt water out in Pearl Harbor beneath that freshwater is up around 24 C, I think. So again, Steve Balls wrote a little chapter in an environmental impact statement with a nice hypothesis, pointing out that offshore from this area, here's Honkahau, here's Kihei where the OTEC is. In the past there was a big submarine slump there, and that's brought a steep underground slope much closer up to the shore in this area. And so back to this diagram, this is in salinity, low salinity cold color, high salinity hot color. Flip them, and if we're talking about temperature, the freshwater lens a little warmer in color, and make this all blue because it's colder. So that would be kind of your temperature thinking for this area. And so this is thermal infrared imagery with Craig and Adam's work, and they found that inshore where groundwater is discharging, you get quite a cold water signature, down on the order of 26, 27. and point out rightly that that's a nice way to go look for coastal groundwater discharge. A wrinkle is that it turns out that it may be the saltwater fraction that's contributing the coldest part of that signature. And that's just kind of an interesting wrinkle, maybe for future thinking. All right, and that's it. Collaborating agencies. Any questions?
Sure, it's plenty.
Yes?
Oh, I have a couple. One was you mentioned turbidity as a tracer, and I wanted to ask, I mean, what it indicates is clear to me, but how do you measure it so that you are certain that nothing else might come in your measurements, like waves and normal? It was right at the beginning, like the first couple of slides.
Turbidity. It's on the instrument. It's a parameter on that multi-parameter instrument that we measure.
Mm-hmm.
And turbidity is a problematic thing to measure. It sounds like you may be expert in it. On the instrument, it comes as a function of the instrument. It's got one of the particular sensors adjusted for it. For my purposes, I'm not an expert in turbidity, so I was happy to use it as a parameter. In my reports, I did display a map of it, just as the other salinity parameter. And I know that people measure it with other types of instrumentation as a lab measurement and so forth. But that's about the extent of my knowledge. So I was operating it pretty much as a stupid instrument and happy to plot it up, but I didn't really have much to say about it, again, not being expert in it. So what were your concerns about it?
Well, no, or what time of the day or what?
Oh, yeah.
What do you have to wait, that there are no storms or anything like that that can?
Yeah, environmental factors. Well, you can see that when you get
Exactly.
this kind of runoff with muddy water, your turbidity through the roof.
Right, but I mean, turbidity can also have just like from waves or external seawater.
Right. You can see here just this little amount of wave action is making this inshore water a little turbid.
Yeah, and that might not be directly related to submarine groundwater discharge, right?
No, not everything that we measured as an indicator of submarine groundwater discharge. The main ones that I would consider with my simplistic subset of measurements would be salinity and temperature as being most diagnostic.
And I have another question. Can we go to the effluent, to the Kihei map, where we look at the effluents?
Sure.
So, towards the, a couple more, a little bit farther where you had, yes, that one. If you look at the most upper red dot on the coastal line, the 17. On the coastal line, the 17 is one
Right
of
here?
the highest. Yeah, you have 16, 15, 16 as a normal measurement. You have 18 somewhere in the middle where you expect the most, and then you have that one 17 next to the 13 where you had your submarine spring, right?
Uh-huh.
And so I'm thinking, do you consider that an outlier right now? Because the 13 of that submarine spring should be pretty reliable, right? Or because between those two you have a really big difference.
Yeah.
Do you relate that back to maybe the topography, because everything is going down slope, and that might be already on the other side, a little
Well,
bit of that?
We think that we're getting towards the margin of the plume here, so you're right, you're getting into an area where you expect a transition. If everything is simple in the world, maybe we don't expect such an abrupt transition, but everything's not so simple in the world, as evidenced by the prominent spring versus not so prominent immediately in other areas. All I can say is these are in algae, they're growing on the bottom and incorporating this, and so they've eaten up what's come their way, and that's the result.
So towards that 13, would you expect already effluents from other parts, not from the wastewater treatment facilities, but from somewhere further now?
That's a very good question. I think maybe not so much for a value of 13, because we've got values of 5 measured on land here. But a very good principle is that whatever, as that water moves and discharges in the coastal sediments, both at the shore, just offshore, and onshore here, this is, the tan here means that this is all sedimentary, the green is basalt. In these environments, the sedimentary environments, you will have populations of bacteria that will start to munch on nitrogen and start to denitrify it. And so a slight heavying from 5 to 6 or 9 or 6 up in this area, maybe that's what you're getting for other nutrient sources besides the plume with a little bit of denitrification. Yeah, that's the kind of thinking you want to go through. I didn't have all that much to say about it because it's really reconnaissance mapping, and I can't get too speculative. But yeah, you can. Those are some of the principles you'd want to pursue with thinking further in the process. Anyone else? Yes.
The concentrations of the 2 drugs seems actually pretty high, you know?
Parts
An
per trillion.
epileptic drug and a sulfone drug. You know, I mean, those are not over the counter.
No, those are
There's a lot of
prescribed.
sick people on Maui, or.
Well,
Does anybody?
no, very good point.
What is the concentration in a tablet, and how many people are taking this?
I don't know, but in USGS, we've got a web page. If you keyword search for USGS emerging contaminants, there are whole study groups back on the mainland that have been looking at this stuff. They first became of interest because you can use them as a tracer like this, in a simple-minded way. But then other aspects are, well, if a treatment plant discharges into a river on the mainland, there's another town downtown that's got a water intake. So, for drinking water. So what are people maybe ingesting that's been discarded farther upstream? So there have been detailed studies of, okay, what goes into the plant, what concentrations go into the plant, what come out of it, what's degraded, what doesn't get degraded. There's a whole bunch of research about that. So if you search for USGS emerging contaminants, you'll get to the web page.
So people have actually looked at going into the plant,
Yeah.
how much the concentrations.
Yeah, people have studied that in depth. I listed that for any of the SGD group. It's listed as one of the reading material entries, that web page.
Okay.
Good question, concentrations. The other thing I wanted to add is we're largely detecting this because it's a centralized municipal wastewater plant that pulls wastewater from a large population. If you're out in a terrain where there are individual cesspools and maybe 20 or 40 households, like up in Kahana or wherever up on the windward side, of that population, you might not have people on those particular drugs. But once you get to a certain size population, you do. The other kind of things, people go in for their medical imaging. They give you something to drink ahead of time. You can detect that in effluents. They're very detectable because they're based on rare earths. And they can be just about like the only source for those kind of rare earths in a particular area. So those are hot with one of our researchers named Larry Barber. So yeah, all sorts of interesting tracers with these pharmaceuticals. They're expensive.
Yeah.
But you can supplement other approaches with a few of them and get some results. Any other questions? Yes, sir.
I think so far the DOH has not found these chemicals in drinking water. Where is it coming up?
I did a round of reconnaissance sampling in 2001 for the Nautical project and got to sample a couple of wells up in central Oahu. And we got a detection or two on, well, let's see, chloroform was found quite pervasively at low concentrations. And and that's thought to be either a disinfection byproduct from chlorinating water with a slight amount of organic matter in it. Or my hypothesis was also that it might be a breakdown result from some of the chlorinated solvents that are known to have contaminated up in that area. As far as these particular pharmaceuticals, I don't think I detected much of that. And there's a recent program where they've gone and sampled for a newer generation of these analytical schedules. And it's not been released yet, but will be soon, I think, to DOH, the results of that sampling. That's up in central Oahu. The hypothesis there is that there's a question, is it safe to reuse wastewater by, say, applying it to agricultural fields in central Oahu? You're worried about what might percolate down to the deep groundwater. So there have been applied studies where they make application plots and apply and then sample in monitor wells and piezometers immediately beneath at shallow depth. But up in central Oahu, there's one treatment plant on Lake Wilson from which water is drawn out into a ditch system and has been used to irrigate agricultural lands up there for decades, since the early part of the 20th century. The treatment plant's probably not been in quite that long, but so there's a natural experiment going on there where those waters have been applied to agricultural lands for decades, at least 60 years or so. And the idea is, well, let's sample a couple wells and see if we detect anything from that type of agricultural application. So there's been a sampling done, and results are just coming in. And being released to Department of Health soon.
But nationwide, these chemicals are not regulated now, or
Question is, are these chemicals regulated? No. Many of them are an unavoidable result of medications we take or things that we wash off with. And there is concern, like I say, particularly in rivers. But no, not regulated, and almost impossible to regulate for me.
very expensive.
Yeah. They're also not very easily removed, some of these. In wastewater treatment, there are certain methods used to remove chemicals of concern. Again, you're talking parts per trillion. It's hard to remove everything from groundwater down to parts per trillion. So again, it's quite a thriving field of study. We've only used it out here in Hawaii as simple-minded tracers, but people are heavily researching the processes and ecological effects and health effects. Some of them there are real ecological concerns. Some of these chemicals can be endocrine disruptors, and there is concern for things like amphibians and fish and various critters with some of these constituents. Again, that's a thriving topic. There's plenty of it online. And again, search for that USGS emerging contaminants website, and you'll find all the research on there that USGS does at least. Any other questions? Yes?
We've kind of gone over, so we're going to meet, the class will meet back with Chip, so I want to let everybody